Now we are going to see about sequencing methods where DNA sequencing is a fundamental technique used in modern biology which is to about study the genes, functions and their interaction. It helps to uncover the genetic basis of the disease and the drug therapies and to monitor people response through those treatments and to shed light in the evolution of the life where the Max and Gilbert method were used early days for the DNA sequencing, but now they are using more technologies. Now we can see about the history of two DNA sequencing methods, where it is started in 1970s, and there were two DNA sequencing methods done directly. One is the Maxim Gilbert sequencing method, and another one is the Sanger chain termination methods. In 1980s, where both Walter Gilbert and Frederick Sanger were awarded for the Nobel Prize in the chemistry for their contribution towards the determination of base sequences in the nucleotides. And Paul Berg received the other half of his research into the nucleic acid biochemistry and through the recombinant DNA. Therefore, the Maxim Gilbert sequencing and the Sanger method comprise the first generation of the sequencing method. Now, how does the Gilbert sequencing method works? It it is a chemical sequencing, but because the chemical reactions rather than DNA and RNA amplifications are the basis of this method. First, we are going to prepare the sample, and next cleavage, then electrophoresis and autoradiography will take place. During the preparation of your sample, the first denaturated single stranded chains and radio labeled as the 5 and usually with the 32 pairs. During the cleavage, the next steps cleaves the DNA and the sequencing get really interesting. And then electrophoresis, the electrophoresis take place, and the autoradiography. Then why it is lost popularity? Because it has a simple principles, came with a lot of troubles, and it has a major drawbacks. First, time consuming, and then supposing that everything went well on the first try, a lot of steps of this method could cause problems. For examples, the steps include radio labeling, the cleavage reactions, preparing the gel, electrophoresis, X-ray films. Then the Sanger sequencing. Sanger sequencing is also known as the chain termination method, were developed by the English biochemist Frederick Sanger at 1977. This method is determined for the DNA sequencing about the nucleotide bases in a piece of DNA, commonly less than 1000 base pairs in length. Sanger sequencing was used in Human Genome Project to determine the sequences of relatively small fragments of the human DNA. These fragments used to assemble the larger DNA fragments eventually in the entire chromosomes. How does this Sanger sequencing work? By the below principles. In Sanger sequencing, a DNA primer complementary to the template DNA used in the starting point of the DNA synthesis. For the deoxynucleotides, triophosphates, and the polymerases extends the primer by adding the complementary DTNP to the template DNA strands. It has usually the several steps. The first step is to double-stranded DNA is to denature into two single-stranded DNA. Then the primer that attach or corresponds to the one end of the sequence, and they both away get attached. Then the polymerase solutions is to get added in the solution. Then the DTNTP is added. Then the DNA synthesis reaction initiate the chain extends until the termination of the nucleotides is randomly incorporated. The results DNA fragments are denatured into ssDNA. The denatured fragments are separated by the gel electrophoresis, and the sequence can be determined. Where these are the steps. First, we have to denature the DNA by heat, then the primer get attached, then we have to add the four polymerase solutions. Then the complementary DNA chains get terminated using the dye. Then denature the ground chains, and the by the electrophoresis we can analyze the results. Next, automated DNA sequencing. Automated DNA sequencing is involves four fluorophores for each of the four nucleotide spaces. Then the resulting fluorescent signal is recorded at a fixed point when DNA passes through the capillary contents during an electrophoric gel. Then the basic specifics label attached to the appropriate in the DTNTP. Each DTNTP is labeled with the different colors. For example, green, blue, yellow, and red. According to the colors, nucleotide may be different. All chains terminated at the adenine will yield green signals, and all chains terminated at the cytosine will yield the blue signals, and so on. Then the reactions based of this chain terminations labeled nucleotide were carried automatically in the capillary pillars. Then the electrophoretic migrations in the DTNTP labeled chains in the gel, and the capillary passes in front of the laser beam. Then the laser introduces the fluorescent signal that is dependent on the specific label representing one of the four nucleotides. Then the sequence is electronically read and recorded and visualized as the alternating peaks of the four colors representing the nucleotides in their sequence positions. This how the automated DNA sequencing works.